not going to be able to tell you anything really specific, especially about steel, which I have sort of a learning barrier there. But I wanted to talk about some of the new things we're finding out about soils. And when I sat down and made a list, I came up with 8 topics of new discoveries. And don't worry, I'm not going there. I narrowed it down to 3 that I think are key areas where there's a lot of new information and a lot of research effort being directed right now. And I did simplify my title a bit. Okay, I have to get oriented here.
Page down, up and down.
This? Where's that? Oh, down there. I never really figured that out. Really obscure.
Oh, sorry.
Okay, there. Okay. Okay. This is different than most of them. Okay. So this is the overview of the 3 topics. So I'm first going to talk about this question of, well, how does organic management affect the soil microbial community? And a lot of interest and speculation in that, and we're actually beginning to be able to ask that question. And then 2 related topics about ultimate microbial habitat, the root-soil interface, and decomposition. Who's in charge? We think of that as a microbial process right now, for the most part. I'll start with my first topic about the microbial community. So there has been this long-held belief in the organic farming community, as well as in researchers that are interested in a more ecologically based farming system, that these practices would somehow change the microbial community composition, and that you would have different species presence, and maybe differences that would sort of cascade into nutrient availability. And, oops, I did not want to go backwards. And so just at the beginning of that sort of question, we now have lots of studies that show differences in organic matter composition and differences in nutrient cycling. And these are really important from a microbial perspective, since they inhabit the organic matter and they carry out the majority of the nutrient cycling. And then more recently, there are several studies that actually get down to and look at the microbial biomass just as a whole and say, well, yes, there's often more decomposers around, and we do find some differences in the enzymes, because the microbes are producing enzymes in order to carry out the process of decomposition or breaking down organic matter. But then if we get to the point of, well, let's look at who's there. Those studies are rare, but they are really we're on this part of the curve, where they're really going up, because the techniques that are available to ask the question about microbial species and then connect that to what they're doing for us in the soil is really at the sort of the early stages of being accessible for this kind of environmental question. So we've been doing a little bit of that work in my lab. And this is Sean Berthog's work. I'm not a microbial molecular biologist, but he is. And we've been doing this work in Pinyan, in a couple of fields out there. And the organic fields are Klaus Martin's fields, shown here. And then we have 2 adjacent conventional fields. So these are on very similar soils. They're essentially the same mixtures of 2 common soil types, Honioy and Lima soils. So we've been doing work in these fields for probably 6 or 8 years now. We started out characterizing the organic matter pools and looking at other processes. Now we're moving on to look at the microbial community in these soils. And I did forget to say at the beginning that if there's any place where you have a question you want me to clarify, or where I've let jargon slip into my talk, then stop me. So when we do these experiments, we're using very small amounts of soil. This is just showing the incubation flask that Sean is using for some of the studies, looking at what the microbes are doing in terms of cycling carbon and nitrogen. And that's one of the challenges, is that we're trying to take information from little bits of soil and then figure out, well, what does it mean for a field? What does it mean in the big scheme of things? And I just want to mention that caveat. So this is how we look at information about the microbial community composition. I'm going to walk you through this. And because I'm not a molecular person, hopefully you'll be able to follow, because I have a really hard time. By the time the molecular biologists get through telling me how they got this information, I'm glaze. So basically what they did is extracted the microbial genetic material from the soil. And so along this axis are all the different species of microbes. And it's controversial about exactly what a species is. And you can see by these clusters, this dimension is how related they are to each other based on their genetics. So we do have some clusters of some related microbial species in this. Okay, and then I have to go over to this side, which doesn't mess up the video. Yeah, so over on this side, we had the information about the fields. We had 2 organic fields, 2 conventional fields. Red is conventional, green is organic. And the color of blue is the abundance, so how many of these species were there. And so in this first area, so this cluster of species, where we don't see a lot of diversity in terms of they're closely related, we find these in all the fields for the most part. So these are, we think of these as the generalists. Basically, organic, conventional doesn't matter. When we move on to this part of the spectrum, you can see a lot of blue down here in the organic fields. We had 3 samples from each field. But we don't see them up here. So this means we have a whole batch of microbial species and groups that are present in the organic fields and absent from the conventional. So this is clearly showing that we have a group of unique species that are present in these organically managed farms. Now, at this point, we don't know which species they are, and we don't really know what they're doing. But this is very interesting. I tried to find some other studies in the literature that sort of were having these kinds of striking results. And there's a study in strawberries that shows some very profound differences. But of course, those systems are fumigated on an annual basis in the conventional system. So that sort of kills everything, and then you start over again. So you would expect to find the difference. I'm not going to spend as much time talking about the fungi. We also looked at the fungi as well as the bacteria. Now, can anybody tell me why we focus on these 2 groups if we're interested in decomposition and carbon and nitrogen cycling? Don't want to put you on the spot. Anyone want to guess? Hazard guess? I mean, it's fun to look at the little critters that can crawl around, and you can actually see them. These guys, we get a bunch of colors and squares. No one's going to venture.
Probably.
Exactly. Yeah, you're right. He said they're important for nutrient release, and they're really up to bat first. So they're the primary decomposers, and they're the ones that really have the enzymes and the ability to break down all these complicated organics. So that's why we all focus on them, even though especially the bacteria, can't really see them. Okay, so for the fungi, we had a lot more species. We do have difference in the actual structure, so the community structure differs by management. But we also had a lot more variability by fields. And I'm just going to point out down here that this field had this whole group that are related, but the other organic fields didn't have this group, nor did the conventional. Here's another group of somewhat related fungi here that were present in this conventional field, but not really present elsewhere. So we had a lot more spatial heterogeneity or field-to-field variation, and not a huge difference just in diversity as a whole, the difference in the species that are present. Okay, so the bottom line of this work. The bacterial and fungal communities are distinct in these organically and conventionally managed fields. There's other studies that use earlier methods that suggest this, but they all have, there's sort of lots of caveats with the methods. We did find these unique bacterial groups in organically managed fields. And fungal groups varied from field to field, so that's going to make it more challenging to study this group because we've got to understand the landscape variability and the management effects. So now we're very interested in looking at the relationship between the species that are present and what's happening in terms of carbon and nitrogen cycling. And we're especially interested in those unique bacterial groups that are present in the organic soils. We really want to know what are they doing there and how did they get there. Are they there because they've been promoted, or are they just being suppressed by maybe some of the chemicals used in conventional? So we're following some of those directions. Okay, I'm going to shift to the second topic, the ultimate microbial habitat, the root-soil interface. I'm just curious, so how many people have heard of the rhizosphere? Is that a couple? Yeah. It's kind of getting out there. Actually, one of the gardening magazines did an article on the rhizosphere last year, and they called me up, and I was kind of surprised. But so the rhizosphere is where roots, meet the soil, and it is a unique habitat. And I think it's really only recently that we're really appreciating how tremendously important it is. So I'm going to step back a minute. Let's think about where microbes live. We all know they're small. I mean, they're just so darn small that even the photographs of them are kind of boring, not very interesting. So if you think about that, well, from a microbial perspective, the soil environment is huge, huge, and really variable. I mean, really, really bad places that you're just going to die. There's those few sweet spots out there. And there are a lot of them in the soil. We know 100 million in a teaspoon, or possibly a billion, so lots. But still, they're really occupying about 1% of the soil volume. So I think that that we tend to think the soil is rich in life, and but really it's more like a sort of a desert with a lot of oases scattered throughout it. So microbial habitats that are really active, where the microbes congregate, are very specific, close to plant roots, aggregates. There's a lot of activity going on there, and then pieces of organic residue. So microbes are actually most abundant in the rhizosphere, which is actually a good thing in terms of our opportunity to manage some of these processes that we're dependent on, since we're dependent on biology. So this is just a nice sort of scanning EM picture of the rhizosphere showing root hairs, fungal hyphae. There's a lot of mucigel, which is some complex carbohydrates secreted by roots, and then microbes also secrete them. Yes?
Is the rhizosphere assumed to be a certain depth?
Well, it's usually defined as the 1 to 3 millimeters of soil, which is, let's see, that's very, very, very small fraction of an inch. But so 1 to 2 millimeters of soil surrounding roots. So it permeates, the soil wherever roots are able to go. Even, if you have a plant that sends roots very deep in the soil, then they'll take the bacteria with them and create a favorable habitat, even in a hostile environment of soil 50 feet deep. There's not too much other organic matter. Yeah, that's a good question. And then you can sort of see the bacteria. They're just sort of, it looks like stuff, little, tiny debris there. Okay, so one thing that makes us unique is this three-way interaction. So we have interaction going both ways between plants, microbes, and the soil environment. So the soil plays a role in shaping the rhizosphere. So does the plant. Microbes interact with each other. They interact with the plant and the soil. So, oops, pointing the wrong way. This makes the rhizosphere a really unique habitat. There's a lot of metabolism going on there, a lot of communication between plants and microbes. And this impact of plants on the microorganisms in the rhizosphere is really on the forefront of research right now. It's been, I think, seriously neglected, but of course we really didn't have the tools to really investigate, how are plants actually farming the microbes? And it's really the microbes, it's similar to our gut. I mean, we're dependent on a whole bunch of bacteria, So here are some of the processes that we now realize are really jointly or collaboratively managed by microbes and plants together in the rhizosphere. Nutrient cycling, soil organic matter formation, plant health, vigor. Microbes secrete certain compounds that sometimes really improve plant health, and aggregation as examples. And so what determines the community that we find in the rhizosphere? Well, the microbes that are already living in the soil, thanks. And that's why we want to know, well, who's out there when we do the kinds of sampling I talked about before. The plant species, but even the crop variety can actually, you can have different communities in the rhizosphere of different varieties of the same crop. A bunch of other things could, but we don't know. I'm going to go quickly through this now since I have 5 minutes. Consequences of the plant's impact and kind of influence on the communities can vary. We all know about pathogens. We know about plants like tomatoes and corn that tend to promote communities that seem to be problematic or cause disease. But there are cases and there are plants that actually foster beneficial communities around their roots. And this is a big question. I mean, why is there this difference? And we see this out in natural plant communities too, that there are plants that foster pathogens, plants that tend to foster beneficials. We don't know why. I'm going to show one experiment quickly about aggregation. We think of that normally as a soil health property. Plants can have a huge influence on it through cultivating certain microbes. This is an experiment that was done with sunflowers, like this, where they inoculated with an aggregate-forming bacteria. So the bacteria produced these gels that helped to form aggregates. And they also had a control. And then they subjected the plants to water stress. And basically they found that the plants that were inoculated with the aggregate-forming bacteria, so it was in their rhizosphere, had increased aggregation and water holding capacity in the soil around the roots, so that layer of soil near the roots. They had improved resistance to water stress. And they also grew faster, so they did better under drought. There are several studies that show this kind of thing. Decomposition. Okay. I'm just going to mainly try to make the point that decomposition is a process that we really need to think of as jointly controlled by plants and microbes, because so much of it happens in the rhizosphere. And this is the first experiment that sort of showed this relationship. It was published in 1986 and kind of neglected. So Marie Clairholm started with sterile soil and a wheat plant growing in it. And she also had a treatment with sterile soil where she inoculated with bacteria. It did a little better. And she had a third treatment where she had sterile soil and she inoculated with bacteria and grazers. Grazers are like protozoans or nematodes that feed on the bacteria. And what she found is that the wheat growing in the soil that had bacteria, decomposers, and had the next trophic level, which are the grazers, was able to take up the most nitrogen and grow the fastest. And so there are now, since then, and I'm not going to talk about it because I'm running out of time, but I'm going to skip through them. There are now many studies that have finally people started really going after this in the late 1990s to try to understand it. And there are now many studies showing that this rhizosphere decomposition that is, also mineralizes nitrogen and releases it from organic matter is something that is very important, and it varies between plant species. Plants can respond to nitrogen shortage or if they need nitrogen by actually ramping this process up. Just to give you an idea of the magnitude of the difference when an experiment was done just looking in the rhizosphere, nitrogen cycling was going on 10 times faster compared to bulk soil in the rhizosphere. Now I'm skipping all this. Oops, sorry. Oops, I do want to show you this picture though, before I go to the conclusions. So we can actually see that sort of trophic cascade that I explained. We can see the rhizosphere, and we can see that grazers are eating bacteria like crazy in this habitat. So this is a photograph of a root in soil, and what they've done is they've put a fluorescent marker in a bacteria, and then grown it in a culture dish, and then put it in, and put some protozoa in. So when the protozoa, we can't really see the bacteria, they're too small, but when the protozoa eat the bacteria, then we can see them, and there are all these red dots here. So this is just showing that these protozoa in the rhizosphere are just gobbling up bacteria, which releases nitrogen as they digest them for the plant. Okay, last slide, my conclusions. So these two last topics really stick together and tell us that we need to change our view of how we think of plants. And that's going to affect how we manage them. We know now plants really are able to select for beneficial microbes in the rhizosphere. We need to understand more about that so that we can incorporate it into our management. I think especially in organic agriculture, this is going to be very important. They're able to create a community complex that's unique to them, even at the level of varieties. And this may explain why some varieties do better than others under certain conditions. It may also explain where some of the conventional varieties that have been selected under high fertilizer conditions don't do well in organic, because we may have changed their ability to communicate with microbes and to foster a rhizosphere community that's beneficial. And then through these collaborations, plants are influencing many of the processes that relate to the soil health properties we normally think of as just really starting from the soil. So we need to think of them as really, sort of a complex or consortium. We've got insects that are associated with the above ground part of the plant, and we've got a whole community that's associated with the roots, and the plant is influencing both of those. And that's well accepted when we think about insect pests and their beneficials. But we really need to think about the below ground world the same way. And thank you. Here's my
When you compare the organic with conventional fields, was the organic management, was there more tillage going on?
There's probably somewhat more tillage because we chose conventional fields that were conventional tillage, and they use herbicides for Roundup Ready.
I'm only asking because of this.
So there's cultivation, yeah, it's plus mark.
The fungal community comparison might be explained by more tillage in the organic. Those communities recover a little slower.
So you mean the fact that there's not greater diversity, but there's differences in structure? Yeah, I mean, I also think that we had only, this stuff is a lot of work, which is why it's going slow, and you have to use small amounts of soil. But I think we need more fields. We need more sites to really interpret fungal. It may not be, biodiversity in itself is not necessarily inherently beneficial. It's who's there. So you can have, increased biodiversity and see no benefits. And that's just because you get, organisms that are doing the same thing. They're not, you don't have diversity in their function. But that's, that's a
One more question for Laurie, and then we'll get.
You'll have to pick, I don't know.
How about over there?
So is,
Brian, you can ask me anytime.
well,
We're going to drive back together.
it was a question for the group.
Oh.
Could you say then that this could be a case for looking into the possibility of inoculating compost to get diverse populations of microbes and nutrients?
Well, the microorganisms that live in compost are going to be different than those in the soil, and I'm not sure about how far we have to go down the inoculation. I'm more for let's select the plant, because a lot of these critters are in soil. So it's all about the plant being able to say, hey, I recognize you, and I'm going to support you somehow. Because in a study with wheat, looking at fields that had wheat for different lengths of time from 5 to 2,000 years in the Middle East, the older wheat fields, they looked at one microbial species, and that species in the fields that had wheat for a longer time were better at fixing nitrogen for the wheat. So they had co-evolved with the crop. All right,
Great,
thanks. Thank you.
So let's get Sam and Anu up and start our general. You can't leave yet.
And now Brian.
And now Brian can.
Well, just a very quick question. That was the crop that was growing in the organic and the conventional fields the same crop?
Oh yes, I'm sorry. Yeah, we used fields where corn was the previous crop, all 4 of those fields.
Oh, go ahead. I'm sorry.
I just want to ask a question. Do you have any idea about how long the exudate coming off of the roots would still interact with the microorganisms? Like, is it